Next speaker is we invite Dr. Lih-Ren Chen. Dr. Lih-Ren Chen got his degree from Davis, and he is the head of radiology in this institute, right?
Thanks, Dr. Chen, and I would like to express my appreciation to FFTC to give me this chance to share this research result in my institute about porcine embryonic stem cells. Embryonic stem cell was first established from the mouse. That's Dr. Evans and Kaufman in 1981, inspired from the nature of the development of the embryo. So they isolate the inner cell mass from the process stage embryo and establish the mouse cell line. This cell line are capable of cell renew, and they can develop into all the cell type of the body. After the establishment of the mouse ear cell line, many knowledge about the embryology, gene modification, genomics study, and even the regeneration study are founded in this kind of cell line. Even the human ear cell line was established in 1998. And why I had to use this animal to replace the well-established cell line from the mouse? Just because in the evolution, we can find that they are very similar from human to the pig. And even in the embryology, we can find that pig and the human, they go through the very similar process. And even in the literature, the behavior of the pig is very similar to human. So, yes, we try to use pig to establish the cell line from this kind of species. By the way, pigs are demonstrated. They are very similar to the human in automotive immunology, research characters, and even the organ size. And also, this kind of embryo has been served as an excellent animal model in many human disease patterns. By the way, because we cannot use humans as a lab animal, so we try to establish the porcine stem cell based on the aesthetic consideration. And also, the most important is that the curiosity and challenge. So we use this pig, use the pig as a lab animal to do this kind of job. In 1991, we tried to mimic the protocol of the mouse and try to establish the porcine ear cell line from the process stage embryo. At this paper, we established the cell line from the C57B6 mouse and produced the Tainan mouse from process injection into the ICR processes. But at that time, the cell line isolated from the ICM of the pig processes do not result in a stable cell line. So I went to UC Davis to do my PhD, and then I used pig process correct from day 6 to day 8 after insemination. Most of them are from early process stage to later hatch process, and I use them as the material to try to establish the cell line using the whole process. And then I established 12 cell lines from the early hatch process and published in 1999. That's the morphology of the process. And in this study, we found that the colony shape of porcine stem cells are much like the human ear cell, straightened. And also they can form the engulfed body, and they can differentiate into many different cells type from the 3' enlarged there. After I back to Taiwan, I used the pig livestock research institute, pig number 1 as the material, and follow the previous study to use the whole process as the material to isolate the cell line. And this morphology, this is the porcine colony maintained on store cells, and this cell line I named it as 215-3, and the cell type of this cell line is very stable. It's around 95% normal cell type. This one is a male cell line. And also they can express the prepotent marker of ear cell, just like the mouse. And when induced to differentiate, they can differentiate into neuron cell lineage and express the cell marker of the neuron lineage. And they also can be induced to differentiate into mature neuron type and express those neuron-specific cell marker. And because they can express the pH cell marker, so we try to use this kind of cell to do cell transplantation then. And some of the difference between human ear cell, porcine ear cell, and the mouse ear cells, and the monkey ear cell, we can find that the expression of the prepotent cell marker of the porcine ear cell we have is much similar to the human stem cell. They do not express SSEA1, but do express SSE3, 4, and TRA160 and TRA181 and F-octo4, and also they have the AP activity, just like the human stem cell, and different from that of the mouse stem cell. Because mouse stem cell is express SSE1, but not SSE3, 4. And from the behavior and character of the porcine stem cell and other stem cell, we can find that the porcine stem cell, they need store as the feeders to be growth normally and maintain their prepotency, just like humans. But the mouse, they can maintain in a feeder-free system if you use the leaf. But in case of pig stem cell, the leaf is useless in our culture system. And as shown in the figures, we can find that the morphology of the porcine ES cell colony is much like the humans and different from the mouse ES cell, the bone-like the shape of bone-like. And we also found that the porcine ES cell, they can form the embryonic body just like other ES cells. And in my previous study, I inject the porcine ES cells into the blood cyst of the different breed. They can form the camera, but no germ-like camera. Because we had a stable porcine stem cell, embryonic stem cells, we try to make them traceable by introduce the GFP gene into the porcine ES cell by electroporation, and then we obtain 3 lines of the GFP class porcine ES cells, and we use one of them to check the expression of pluripotent cell marker. We found that even they have been introduced GFP gene, but they still can express the normal pluripotent cell markers, and also they can form the embryonic body. And if we direct them to differentiate, they can differentiate into the neuron and the heart muscle cell lineage, and still carry the GFP gene and express the GFP into the cell. So then we application those kind of cells to human disease model. In this paper, we used the porcine ES cell of different differentiation stage and try to cure the human Parkinson's disease. And when we use the rat as a model, because the rat model is the standard model. Firstly, we destroy the destroy the brain area responsible the control of motility. And then we inject the porcine stem cell undifferentiated and direct differentiated for 12 days and 18 days. And then we trace the cells with IVIS system. We found that in the experiment period of 3 months, we can still find the GFP marker of the stem cell transfer, and analyzed by the auto-loader system. We found that if the mouse model was transferred with 18-day neuron precursors, the a systematic movement will be useful. The next disease model we try to use our porcine stem cell is spinal cord injury, and we destroy the spinal on the impact the area with New York impactor, and then we transfer stem cells and trace the viability of the cell into the inner rate. And the results show that the BBB scale of the animal was highly improved with of the rate transfer with day 18 differentiated porcine stem cell. Also, we used the porcine stem cell to try to improve the periodontal vegetation effect of the pig. So this kind of transplantation is the auto transplantation. And the result, after we destroy the molar, the premolar, the second premolar, and transfer the collagen with stem cell, we found that the recovery of the molar was improved in case of the death of the wound and the attached level of the teeth. So in our case, we found that the golden standard of the embryonic stem cell have been almost fulfilled for the putative porcine ear cell and porcine ear cell with GFP marker. So this kind of the result, we can say that the porcine ear cell has been established in our lab. Also, the cell share many similar character and behavior of the human cell. So this kind of cell line is very useful in biomedical research and the regeneration study in case of a human model. And also the theoretical potential of these cells has been demonstrated in the regeneration medical study in the rat model in case of the Parkinson's disease and the spinal cord injury, and also in the pig model of periodontitis. And I think based on this kind of result, the first study of the porcine stem cell might directly, if we can do xenotransplantation directly to the human cell, or it directly benefit the research because we can use this kind of cell doing disease model study, and this research will benefit the research and the development of regeneration medicine in humans. I have to say thanks to all the colleagues in my institute and Dr. Xie in National Chung-Hsing University and our cooperation partner in the Taiwan University and who studies Tsai General Hospital. Thank you for your attention.
Thanks for your lecture. Any questions? Now open for questions. Is there any question? No question. Yeah, you did it very well. I think in the coming future we will apply this type of reviewing. After you use the cell early. Thanks a lot. So, we will also here we cut another 6 今后。
Okay, thank you very much. There's one question, sorry. Can you please identify yourself first?
Okay. My name is Tien from India. Depending on the morphology, your ES cell is different from the Takashi because one is flat and one is bony. And especially the cell marker, the stimulation is similar SE1 or 4. But the Japna is said is 1 and 4, both expressed in his stem cell. So how your opinion is already differentiated in both of one, both of your DSA, some kind of mechanism is in
Okay, I don't know. I'll say I don't know first, but based on the presentation of the Haraguchi, I can find that in the early stage, their morphology or their stem cells are flattened, right? Before the addition of the inhibitor, they are flattened. So I think maybe flattened colony is the original one. It's the very original one of the porcine ESL. And because my ESL line has been maintained from 1998 until now, and they have been cultured over 300 passages and still maintain this kind of morphology. Since maybe this kind of morphology is a typical one. And also I have read other paper, other reports say that SSE1 expression has been identified in some porcine ES cell line. But I don't know. I don't even found the expression of SSE1 messenger RNA in my porcine cell line. So this, in case of this kind of SSE1 expression, I don't know what happened between our cell lines.
So the answer is simple. There is no reliable marker, and this report from some group SSEA1 and SSEA3 are positive. And of course, this is a difference of the genetic background. Actually, nobody knows why the result is different in the researcher, but it is true. Well, in my opinion, the expression pattern of glycoprotein, such as cell surface marker, such as SSEA1, 3, and 4, and the relation to the morphology shape, in my opinion, there is no relation. Yeah. So I didn't check the TLRI 61 and 81 in our established line, but that he showed that positive, right? So there may be a positive in our cell line, maybe or not, I don't know.
Both cells, some kind of, some level differentiation already happened.
Yeah, possible.
to say because during the in vitro condition, not the cell, even the cell in one colony, they have different state of differentiation. Yeah, I think yes.
No more questions.
Thank you very much. Thank you very much. Let's give a big hand to our speaker, Dr. Tien, and to our moderator,